Hey everybody, and welcome to chapter 11. This will be on blood, and your lab this week will also be on blood typing. So this will be the lecture, the recorded lecture that we would have had in class for Tuesday. So here are the listing of the functions of blood. It works to transport gases, oxygen, carbon dioxide, nutrients, waste products, hormones, the transport of processed molecules, regulatory molecules such as hormones. We covered that last time. Blood helps to regulate pH and osmosis, the maintenance of body temperature, protection against any foreign substances. Your white blood cells are all of your immune system response cells, and also helps with clot formation. If there's ever an instance when you're losing blood, blood will go to that site, and the platelets within the blood will help to stop the blood from leaving the body as a clot. So, what is blood composed of? It's composed of plasma, which makes up about 55% of total blood. This is a pale yellow liquid that surrounds all of the cells. Plasma is mostly water with a little bit of proteins and other electrolytes. And then the formed elements of blood are all of the cells and cell fragments. And the formed elements make up about 45% of the total blood. You have your erythrocytes, which are the red blood cells. The leukocytes are the white blood cells. And the thrombocytes are the cells which will go on and make the clotting factors. Plasma proteins, so these are proteins found in the plasma, so not the red blood cells. Albumin is the most abundant plasma protein, and it helps maintain water balance. The globulins help with the immune system, and the fibrinogen aid in clot formation. So here's a look at the composition of blood. So if someone were to take a blood sample of you in a clinic or a hospital setting, they would take the sample in a vial and then centrifuge it, which means spin it extremely quickly, and the blood would form its components as shown here. The plasma would float to the top. The buffy coat is made up all of the um, platelets and white blood cells, and then the formed elements, the red part of the sample, would include all the red blood cells at the bottom. Here just shows you what types of proteins are found in the proteins, what other solutes are located in the plasma, ions, nutrients, waste products, gases. And then here in that buffy coat, we can see our formed elements, the platelets and white blood cells. And we'll go through the 5 different types of white blood cells in a couple slides. Hematopoiesis is the process that produces the formed elements. And in a developing fetus, hematopoiesis occurs in several tissues throughout the body: the liver, the thymus, the spleen, the lymph nodes, and the red bone marrow. And after birth, hematopoiesis is confined primarily to the red bone marrow in your bones, but some white blood cells are produced in certain lymphatic tissues. All your formed elements of your blood, and again, when we say formed elements, we mean all the blood cells and fragments, so pretty much anything other than the plasma, are derived from a single population of cells called stem cells or hematocytoblasts. And these stem cells will differentiate to give rise to different cell lines, each of which ends with the formation of a particular type of formed element. And this just is a chart showing that all of these formed element cells, whether they are a red blood cell, a platelet, or any of the 5 types of white blood cells, will all originate with one type of stem cell called the hematocytoblast, and through differentiation will form in the particular type of cell. Erythrocytes is just another word for red blood cells. We often abbreviate those RBC. It's a disc shaped with thick edges. The nucleus is lost during development, so this is the only cell in your body that has no nucleus, and that is because its function is to be packed as much as possible with oxygen. So there's no space for a nucleus. The only function of erythrocytes is to be packed and transport oxygen to tissues. Their lifespan is about 120 days, and then they will be broken down in the spleen, and parts of the red blood cells will be further used to create more red blood cells. Hemoglobin is the main component of erythrocytes that transports oxygen. And each of these globin proteins is attached to a heme molecule, which contains an iron atom. Oxygen will then bind to the iron, and oxyhemoglobin is just the word for hemoglobin with an oxygen attached to it. So here's a look at hemoglobin. It's kind of these 4 subunit protein molecules that are formed as one hemoglobin molecule. There's a heme or iron group in the middle, and that is what the oxygen will attach to. So one hemoglobin can attach to 4 oxygen molecules. Production of erythrocytes. So when there's decreased levels of oxygen, that causes your kidneys to increase production of the hormone erythropoietin. And erythropoietin, that specific hormone, will stimulate your red bone marrow to produce more erythrocytes. And then the increased erythrocytes, because they can hold more oxygen and deliver them to tissues, that will in turn increase blood oxygen levels. So here's a look at red blood cell production and how decreased blood oxygen will tell your kidneys to increase erythropoiesis, which will increase red blood cell activity from your red bone marrow, and that will in turn increase oxygen. The fate of old erythrocytes. So erythrocytes only last for about 120 days. Old red blood cells, again, red blood cells equals erythrocytes. Those are removed from blood by macrophages in your spleen and your liver. Hemoglobin is broken down, and globin is broken down into amino acids. Hemoglobin's iron will be recycled, and the heme group is converted to a bilirubin. Bilirubin is what your liver takes up and releases it into the small intestine, and that is used as a part of bile to digest fats in your small intestine. So this is a look at hemoglobin breakdown. I'll let you guys read through these steps on your own, so you can pause the lecture to read through them now. And then we'll go on to leukocytes. Leukocytes are the white blood cells. So this is part of the formed elements, a cell type in your blood. These leukocytes, these cells lack hemoglobin. They're much larger than erythrocytes, your red blood cells. They will contain a nucleus, and their functions are to fight off infections and remove dead cells and debris by phagocytosis, which is kind of the eating up or engulfing of them for removal. You have 5 different types of leukocytes, and we divide them into whether they are granulocytes or agranulocytes. And granulocytes will contain specific granules, and these will include your neutrophils, the eosinophils, and the basophils. Your neutrophils are the most common. They will remain in your blood for 10 to 12 hours, and then they'll move to your tissues, and these are phagocytes. Eosinophils help to reduce inflammation. They will destroy parasites. And basophils are the least common type of leukocyte. They will release histamine and heparin, which are important in the immune response. Agranulocytes then are the second type of leukocytes, and these will have no specific granules to them. They'll be monocytes or lymphocytes. Monocytes are the largest. They will produce macrophages, and lymphocytes are a part of the immune response. They will produce your T cells and B cells, which will have different functions. And lymphocytes are important because they will lead to the production of antibodies. And this is a look at the 5 types of leukocytes under a microscope. And the monocytes, so these ones on the bottom are the agranulocytes. So they don't have the granules in them, as you can see in these top 3. And these are just going over neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Platelets then are minute fragments of cells. They each consist of a small amount of cytoplasm surrounded by the cell membrane. They are produced in the red bone marrow from large cells called a megakaryocyte. Small fragments will break off from those megakaryocytes and enter the blood as platelets, and platelets play a very important role in preventing blood loss. So when a blood vessel is damaged due to a cut in the skin or anywhere in the body, blood can leak into other tissues and disrupt more normal function. So blood that is lost must be replaced by the production of new blood or by a transfusion. A vascular spasm is a temporary constriction of the blood vessel to try to keep the blood from leaking out. A platelet plug can seal up small breaks in blood vessels, and blood clotting, coagulation, is the actual act of these platelets to try to form a clot or a plug in a way in the blood itself to try to keep anything from leaking. So a vascular spasm, it's an immediate but temporary constriction of a blood vessel. Smooth muscle in the blood vessel wall will contract, and this constriction can close the small vessels completely to stop blood flow through them. This type of spasm is stimulated by chemicals released by cells of the damaged blood vessel wall. Then a platelet club is very important in maintaining the integrity of the damaged blood vessels. The platelet plug can be described as a series of steps. Platelet adhesion occurs first when platelets will stick to the exposed collagen in a damaged blood vessel wall. And after the platelets adhere to the collagen, they will become activated, change shape, release chemicals. And then in platelet aggregation, fibrinogen will form bridges between these receptors of numerous platelets, which will result in a plug to try to stop the blood from leaking. And this takes you through the steps of the platelet plug formation. Then blood clotting is can be transformed from a liquid to a gel, and a clot is just a network of thread-like proteins called fibrin that will trap blood cells in fluid. A clot will depend on different clotting factors like the proteins in your plasma. They'll only be activated following an injury. They're made in the liver and they require vitamin K. So after an injury, if you lose a lot of blood, a blood clot will try to go to that site to completely stop. Um, Basically, it'll completely change blood into a gel to try to stop blood from flowing or leaving the body or going into tissue. Steps in clot formation. So any sort of injury causes inactive clotting factors to become activated. Prothrombinase, which is a clotting factor enzyme, it acts upon prothrombin, which will be switched to its active form, thrombin. Thrombin will then activate fibrinogen into its active form, fibrin, and fibrin is what will form that network that traps blood into clots. And these are the three different stages of clot formation. Again, I'll let you guys kind of read these on your own here. Clot formation control, it's controlled by anticoagulants, which will prevent clots from forming. Heparin and antithrombin are two types of anticoagulants. Injury will cause enough clotting factors to be activated so that anticoagulants cannot work in that particular area of the body. So you'll notice here, an anticoagulant will try to prevent clots from forming, but injury or extreme stress to the body will overact or supersede the action of those anticoagulants just in order to try to help the body stop that blood loss. Clot retraction is the condensing of a clot. Serum is the plasma squeezed out of the clot, and this will help enhance the healing. Fibrinolysis, so this is the splitting of the fibrin, is just the process of dissolving that clot. The plasminogen, which is a plasma protein, will break down the clot into fibrin. And this is a look at fibrinolysis, which is just a fancy word for saying the splitting up of a blood clot. Blood grouping, so injury or surgery can lead to a blood transfusion. The transfusion reactions or agglutination is a clumping of blood cells. So anytime a blood transfusion goes bad, we call it an agglutination, where the blood cells will clump because they won't interact well together. They do this because all of your red blood cells have antigens, which are molecules on the surface of them. And antibodies are proteins in the plasma which will counteract any antigen that's foreign to it. And your blood groups, such as type A, type B, type O, they are all named according to the antigen or the specific molecules that are on the surface of the erythrocytes. And if you are type A blood, you will have antibodies already in your plasma against type B blood because you only want to interact with other type A blood types. So the ABO blood groups, this is probably one of the most confusing things in all of anatomy and physiology. I still have physiology students who are still confused at this. So if this confuses you, don't get down. Just try to spend some time studying it and then come back to it later. So hopefully, if you don't know your blood type, that's fine. Donate blood, and then they'll tell you what your blood type is. But in your ABO blood group system, there are two types of antigens that may appear on the surface of red blood cells. They are either type A antigen or type B antigen. And again, these are molecules that are on the surface of your red blood cells. And if you have type A blood, you have the type A antigen on your red blood cells. And if you're type B blood, you have the type B antigen on the surface of your cells. It's as simple as that. Um, if you're type AB blood, you have both types of antigens. You have both A and B. If you're type O blood, you don't have either. So type O blood has neither A or B antigens. And the types of antigens found on the surface of your red blood cells will be genetically determined. So this is passed down through your parents, what blood type you will have. So as along with having antigens, which will determine your blood type, we all have antibodies against foreign antigens that are present in the plasma of our blood. So, for example, if you're type A blood, you'll have antibodies against B. So, we call them anti B antibodies. They'll act against type B antigens. Plasma from type B blood contains anti A antibodies, which act against type A antigens. And that's because your blood just wants to interact with the same type of cells with the same types of antigens. So, you will have antibodies against opposite antigens in your blood plasma. Type AB blood plasma has neither type of antibody because if you're type AB blood, what are the antigens on your red blood cells? You have A and B antigens. So you won't create antibodies trying to destroy these antigens because you have both on your red blood cells. So type AB blood has neither type of antibody. But if you're type O blood, type O blood people have neither antigen on their red blood cell surface. So if you're type O blood, your plasma will have both antibodies against A and against B. In Caucasians, this is just the distribution of blood type in the United States, and it's kind of interesting to see across race the different blood types if you're interested in that. The distribution is type O 47%, followed by type A. In general, around the world, type O is the most common, following type A, type B. And this just takes you through the different percentages. So this is a great chart, and I love this. I use this in all my classes. This is a visual representation of the antigens on the surface of a red blood cell. And again, those antigens give the blood a specific name or a specific blood group. So if you're type A blood, you have the A antigen, and your blood plasma will make antibodies against the B antigen. If you're type B blood, you have the B antigen on the surface of your red blood cells, these little pointy triangles, and your blood plasma will make antibodies against the A antigen. If you're type AB blood, you'll have both types of antigens. And because you have both antigens on your own red blood cells, you're not going to create any antibodies because you don't want to destroy your own red blood cells. And if you're type O, you don't have either antigen, so you will create antibodies to both antigen. Agglutination reaction. So this is what occurs. This is a great visual representation of what agglutination is. Agglutination just means a bad blood transfusion, and agglutination means clumping of the red blood cells. So if you are a type A blood and you're donating blood, and you're donating to another type A person, the antibodies on that person, the anti-B antibodies in the blood of the recipient have this kind of triangle shape to them. They cannot grab hold of your antigens because a circle doesn't fit into a triangle. So there's no agglutination, there's no clumping. However, if you are type A blood, you have the A antigens, these circular sphere structures, and you donate to a type B person, the type B blood type will have antibodies against A, and these antibodies are specifically designed to grab hold of those A antigens and to clump or grab hold of them and clump those cells together. And that's what agglutination is. And if your blood cells agglutinate due to a bad transfusion, they basically become destroyed because they're clumping together and they're not able to travel through your bloodstream to deliver oxygen to the tissues it needs. O is a universal donor because they have no antigens. So if you're type O, specifically type O negative, and I think we'll talk about what positive and negative means in a little bit. O is the universal donor because they have no antigens. Type A can receive blood from type A and O because O is the universal donor. Type B can receive blood from B and O, and type AB can receive blood from A, B, and AB blood. So we call type B blood the universal recipient, and type O can only receive blood from other type O people. So where does the positive and negative come in? Well, there's an extra antigen, and we call this the Rh factor. Rh positive means you have these Rh antigens. 95 to 85% of the population is Rh positive. My blood type is A positive. Antibodies only develop if an Rh negative person is exposed to Rh positive blood. So, for example, if you are A negative, you will only create antibodies against that Rh factor if you're exposed to Rh positive blood. This can be a little dangerous in pregnancy. So it can be exposed, someone can be exposed to Rh positive blood by transfusion or from a mother to a fetus. So, for example, if a mother is negative and a fetus is positive, the mother can be exposed to that positive blood if fetal blood will leak through the placenta and mix with the mother's blood. The first time this occurs, the mother's blood will produce antibodies against those antigens. That first pregnancy, that fetus will be born normally and be fine. But any repeated mixing of blood can cause a reaction. So if the mother gets pregnant again and has another Rh positive fetus, she now has antibodies build up in her blood from being exposed to it from a previous pregnancy. And that can affect the blood of the fetus if the blood mixes again. So this will cause what we call hemolytic disease of the newborn, which occurs when the mother produces antibodies against that positive antigen. It will cross the placenta and agglutination will occur. So the blood cells of the fetus will clump together, but they will also split apart. So fetal erythrocytes will split apart. This can be fatal to the fetus. It will be prevented if a mother is treated with what we call RhoGAM, which contains antibodies against the Rh antigens itself. So it can be treated. And this kind of takes you through a summary of what will happen. Again, this only occurs in the first pregnancy if an Rh negative mother creates antibodies against an Rh positive fetus's blood. That first fetus or first pregnancy will be fine, but subsequent pregnancies could be in danger because the mother now has those antibodies against that Rh factor, which could harm the fetus blood. Diagnostic blood tests. This is what many clinics use to determine blood numbers, or especially white blood cells, red blood cells. The complete blood count is what we call, this provides information such as red blood cell count, hemoglobin, hematocrit, and white blood cell count. The hematocrit is the percentage of total blood volume composed of your red blood cells, and hemoglobin determines the amount of hemoglobin. If it's too low, it could indicate anemia. So this is the hematocrit. They will withdraw blood into a hematocrit tube, usually by a pinprick, and then they will centrifuge it, spin it down, and after they spin it down extremely quickly, The blood sample will separate into its component parts, and then they are able to determine the hematocrit scale. The white blood cell and platelets will form the buffy coat, the plasma will form the part above it, and the red blood cells will sink to the bottom, so they're able to determine blood cell counts. Prothrombin time is the time it takes for blood to begin clotting. This is especially important in the elderly if they're on blood thinners to try to decrease the risk of heart attack or blood clots in the heart. White blood cell count is the total number of white blood cells. White blood cell differential count determines the percentage of each of the 5 kinds of leukocytes. And these are the normal percentages of the 5 different types of leukocytes in your blood. Again, neutrophils are the most common, going down all the way to basophils. Leukopenia is a low white blood cell count. It's caused by radiation, chemotherapy drugs, tumors, and viral infections. And that's because chemotherapy and radiation tries to kill off all the cancer cells. And in doing so, they kill off other white blood cell counts. Leukocytosis is a high white blood cell count, and it's usually caused by infections and leukemia. If you have a high white blood cell count, it means your white blood cells are trying to fight off an infection. So that can be indicative of a even more serious disease. And that's the end of this slideshow for blood. Thanks for listening, guys, and I hope you have a great day.